welcome to our journal club session. We are very happy to be the first one to holding this session. Today, Jolene, I and together with Dr. Harburg will introduce the Boeing trial design in phase one trials and the extension of backfilling nested in Boeing trials. So the ideas is basically from the paper published in 2024. So first, I will use the project. I will briefly introduce the project Optimus. As we know that the FDA has launched the project Optimus in 2021. The project is aimed to improve the dose finding strategies for oncology drugs, and based on its focus on not only the efficacy but also the safety and the tolerability as well. And the project have hugely transforming the phase one trial design since the recommendation of the project. So these are a table I searched online made by Praxel, and they conclude that how the project has reshaped the phase one trial. So we can see that under the old framework, the phase one trials focus on find exactly the MTD, which stand for maximum tolerated dose. But under the recommendation of Project Optimus, we are trying to identify a range of active doses. And under the old framework, we use static rule-based 3+3 dose escalation design, which was very traditional trial design in phase one trial, and use the results from the 3+3 to to make the selection for phase two recommend phase two dose, and that's the main idea and the target of the phase one trial. But under the Project Optimus, they recommend to use more flexible and efficient trial design to find. They recommend to expanding the cohort to find the optimal dose. That means that the cohort we may not limit it to only 3 or 6 patient. We can have more patient in one trial, and which gives us more evidence for a selection of the RP2D. So the Boeing trial design we'd like to introduce today is an improved dose escalation method that it did align with some of the aspects of the Project Optimus, particularly in reducing the unnecessary exposure to high toxicity doses, but it still focused primarily on MTD selection. So at least the other two trial methods that can also be used in the phase one dose finding trials for your interest. So we have this continuous reassessment method as a Bayesian adaptive design, and we also have MTPI design, which stands for the modified toxicity probability interval design. So as a background, I will introduce the 3+3 design as the old method we use very frequently in under the old frame. So the 3+3 design have the feature of it's very simple, and it has poor operating characteristics, and also it's a lack of the flexibility. So in the 3+3 design, we only use 1/3, 2/3, or 1/6, 2/6 as the checkpoint to determine whether we escalate or de-escalate the dose. So it's kind of the less adaptive. But the Boeing trial design enables we can use the information data from all the previously treated patients So we are not the message will not limited to only limited patients in the cohort. So that's the main difference between the Boeing trial design and the 3+3. This page tells about like the main ideas of the Boeing. So here the IN stand for the interval lambda E and lambda D, and that's the key point of this kind of this trial design. So the interval was manually created by us. Mm, that and the lambda E and lambda D was defined as if we in our phase one process, we will observe whether the patient will experience the DLT event, which means the dose limiting toxicity events, which it's a serious adverse events will happen in phase one. So we use the rate of this event to decide whether we will escalate the dose level or de-escalate dose level, and to find ideal level, which was the MTD, the maximum tolerated dose. And that's the main idea of the phase one. So here we set the interval, and the lambda E means that if the observed rate is lower than lambda E, so we will choose to escalate the dose level. And if the event rate is higher than lambda D, the upper boundary of the interval, we will choose to de-escalate the dose level. And if the probability is between the lambda E and lambda D, so we'll choose to stay at the current dose and use the message to see whether the future patient to consider more patients in the future study. So that's the basic process of the Boeing design. So first we will treat the first patient or the first cohort of patients. The cohort is always set as the three. Like we said, we treat the first three patients with the lowest dose. And then we decide to whether de-escalate or escalate this dose. So here we'll use the parameter P to calculate the observed DLT rate, and use these parameters to make the comparison with our interval. And we will repeat these steps until the maximum sample size is reached. And when the maximum sample size was reached, the trial is complete, and we will select the dose whose isotonic estimates of toxicity rate is closest to the target toxicity probability as MTD. So the target toxicity probability, which stands for phi, was set by clinician before the trial. So they may have a target like how much these drugs, how much is it for this drug, and that was decided by the clinician. This page shows the mathematics series statistical features behind the Boeing trial design. So we define these these parameters. Since I have introduced that the phi is the target probability and the p is observed probability, and then we also have phi 1 and phi 2 as the boundary of the probability, which corresponding to our lambda E and lambda D. So the author recommend the phi 1 equal to 0.65 and phi 2 equal to 1.4. So the theory behind the trial design is that we have made three hypotheses that whether to retain the current dose or escalate or de-escalate it. So since it's the hypothesis, so there there exists the incorrect decision rate. So we sometimes we may make the incorrect decision. So we use this alpha to calculate the sum of all the three possible incorrect decision rates. So this one corresponding to the means that if we made the false decision to not retain the current dose when we need to retain the given the hypothesis that we need to retain on the current dose. So the other two are very similar. So by minimizing these incorrect decision rates, so we will use the CDF of the binomial distribution to calculate the parameters, the boundaries we will need to use in our trial, the boundaries and the interval. So here, since these equations are from different papers, so the lambda 1j corresponding to the lambda e and lambda 2j corresponds to lambda d. So we can see that these are the equations, like how we define and calculate the lambda e. We can simply use our target probability and the recombinant probability to obtain an interval, which we'll use to make the decision. So that's the main idea of the Bayesian trials. So are there any questions about that? Okay, so I will keep going. They have an application on their website, so we can simply type in all the parameters we may use in this application size, like the number of total doses and the starting dose level, the cohort size is 3, and we set the number of cohort 10. So it means that we will the total sample size is 30. So and we set the target probability as 0.25 and keeps the other setting as the default. And we can obtain the decision table and the flow chart, the decision table and the flow chart. The nurses, a clinician and the nurses who may not have strong biostatistician background, they can simply use this decision table and the flow chart to decide whether to which level will the patient will be assigned to. So we can see that we'll start at pre-specific starting dose. We'll treat first the 3 patients and to deciding whether the stopping rule was met, and to use this interval boundaries to decide whether we escalate the dose or retain the current dose or de-escalate it. So the decision table is shown here. It was we can easily see that the first column, the first row is the number of patients we treated, and we will make the decision if the DLT is exactly the number, whether we will make the decisions. Yeah. So after the introduction of Bayesian, the author they are promoting the backfilling method massively in Bayesian these years, since there has been increasing interest in backfilling patients during the dose escalation. So the benefits of the backfilling will be we can collect more safety and activity data at multiple dose levels, which under the recommendation of the Project Optimus. And this message can inform us the selection of the RP2D. It can also shorten the timeline of the dose optimization. So as far as I know, the backfilling method haven't been widely used in industry since it was published in 2024, but the Boeing method has been widely used. And the first paper for Boeing, I think that was published in 2014. Yeah, so I'd like to introduce this method here. It's also to hearing from the videos from you to whether like is it feasible or makes sense in the future future phase 1 trial design. And so here is the plot of simulation trials for the backfilling. So we can see that the circle one means the normal dose escalation patients as the cohort of 3. So we can see that the square one was the backfilling patient. So when we conduct the dose escalation process, we can at the same time recruit patients and assign them to the backfilling dose level, which was proved safe and effective to the tumor. And since the normal process may have some waiting time, since that the error means that the number 9 patient is still under the assessment of the result. So during the assessment time, we can assign more patients, assign the 10 to 13 patients to the backfilling dose, and they will also provide us safety and activity data. So we'll also use the backfilling data to decide whether to escalate or de-escalate the dose. Yeah, so here's the 2 conditions that we may need to consider for whether the dose B is eligible for backfilling. So since as I told, the dose need to be proved safe and effective. So our backfilling dose may be lower than the current dose for dose escalation. And we'll at least have one response observed lower or at dose B. The response means that the drug will have some like clinical response on the tumor. So which means that it should be effective. So there's some different between the backfilling process and the normal Boeing trial design. So there may be some conflation between the backfilling patients and the dose escalation group. Since just as I told, they both provide the data and the additional data obtained from the backfilling group, they may lead us to make different escalate or de-escalate decision compared with the dose escalation, the normal Boeing group. For example, we may observe the DLT rate observed at the backfilling dose, that the backfilling dose may be higher than the DLT rate observed at the current dose. So here's the detail of the confliction, and we can see that if the backfilling group's decision is to stay at the current dose, but the current dose for escalation the decision may be escalation that exists that shows the confliction. Similarly, if the decision for backfilling group is de-escalate, but the current dose was escalation, there is a confliction. So it is worth mention that if the decision for both subgroup are de-escalate, there's also the conflict. It is easy to imagine that we have three groups, the lowest one and the higher one and the highest one. Since the highest one is the escalation group and the higher one, the middle one is the backfilling group. So our backfilling group have the decision to de-escalate. So the decision is to de-escalate from the middle one to the lowest one. But the current dose, the escalation group, the decision is to de-escalate from the highest one to the middle one. So that should be the confliction between the two groups, since they both, if they both have the decision for de-escalation. Yeah, so I will ask that, are there any problems or questions? You can feel free to type in or just interrupt me at any time. Okay, so I will keep on. So I have just introduced the confliction we may meet in the trials, but so how do we solve the confliction? So we use this pool DLT rates. We pull the data from the patient from the backfilling group and we pull the backfilling group data and the dose escalation group data. We put it together and calculate the pool probability of the DLT, and also use this pool build probability to compare with our Bayes interval to make the decision. So we will include the total number of patients experience DLT from the dose B star. B star here means the backfilling group, 2K. So K here means the higher dose, which experience the dose escalation group. And so this is how we address the problem of confliction. So here's the three endpoints we may have in both in the Bayes trials and the backfilling nested Bayes trials. So the first endpoint is for the Bayes for the backfilling. So the dose B is close for backfilling if the total number of patients treated at this dose. So we include the backfilling and the dose escalation process together. If the total patient reached the end cap, we will stop backfilling here. We will close backfilling for this dose. And we also have a maximum total number of patients in the dose escalation process. The third endpoint is the total number of patients at the current dose. We will set this delimitation. Excuse me, Dr. James, do you have any? Okay. Dr. Jebs, do you have any views?
No, just keep going.
Okay, got you. So the third endpoint is the total number of patients at current dose. We include the backfilling and dose escalation together. And this one is set of the total trials, not only for the backfilling. So when we complete the whole trials, we will select the MTD using the same method, the isotonic regression as the Bayesian trials, but we'll base both on the data from the backfilling patient and the dose escalation patient. Here it's worth mentioning that we will always set the end cap for backfilling limitation more than end stop, since we may desire more patient in the backfilling process, and since the dose was proved safe and effective. Yeah. So here is assign rails when we recruit patients. Like when we have a patient, which group we will assign them to, whether so if the current cohort is the dose escalation cohort, and the cohort haven't been completely filled, so we will assign the patient to the normal dose escalation process. Otherwise, we will assign them to the highest dose open for backfilling. That's because the higher dose may be more likely to be more closer to our RP2D, and it's have more like greater efficacy. Yeah, so I will stop here for waiting for any like questions or problems, and Jolene will introduce our simulation study. As a simulation backfilling Bayesian trial design, which can make us better understanding the trials. Yeah. And Johnny will also compare the backfilling nested Bayesian with the normal Bayesian, with some operating characteristics that may prove that the backfilling process have some benefit. Yeah.
Thank you. Can I share my screen?
Okay, let me stop first.
Yeah, can anyone see my screen?
Yeah, we can see.
Thank you.
Yes, but you need to put it on presentation.
Right. Okay, so I'll introduce a hypothetical trial example of backfilling Bayesian with 3 patients per cohort. And you can see in this graph, the yellow color means DLT happened and blue color means no DLT happened. And the circle means dose escalation patient and square means the backfilling patients. And if you see a R above circle or square, it means a clinical response for a solid tumor. And the study continued until the sample size for dose escalation is reached to 30, or the numbers of patients treated at the current dose reached 9. For example, like here, after the patient 29 was enrolled and the number of patients at dose level 2 has reached to 9, so the study stopped. And for backfilling, a dose is closed for backfilling when the total number of patients treated at this level reached 12, which is not happened in this trial. And let's see the escalation boundary for this trial. It was 0.196, and if the observed DLT rate at the current dose level is less than or equal to this boundary, the dose will escalate to the next higher dose level. And the de-escalation boundary was 0.297 if the observed DLT rate at the current dose level is greater or equal to this boundary, the dose is considered too toxic. So the trial should be de-escalated to a lower dose. Okay, let's see how this trial did the dose escalation and backfilling. See here, like at dose level 1, and there is no DLT happen in the first cohort of 3 patients, so the dose escalate to the level 2. Also, there's no DLT happens and only 1 response were observed at dose level 2. So therefore, the dose was escalated to the level 3, and dose level 2 was open for backfilling. Accordingly, patients 7 to 9 cohort 3 here were assigned to dose level 3. And before the cohort 3 had completed their DLT assignment, an additional patient became available, patient 10 to patient 13, and they were backfilled to dose level 2. And by the arrival of patient 14, the cohort 3 has completed assessment with no DLT, and simultaneously at dose 2 backfill, patients 10 and 11 completed assignment with overall rate of 1 out of 5 DLT. So there will be a conflict arise for the patient 14 between those level 3 and level 4. And this is a stay or escalate conflict. The DLT rate in dose level 2 is 1 out of 1. And DLT rate for cohort 3 is 0 out of 3. So when the DLT rate is pooled, which is 1 out of 8, which is lower than our escalation boundary. So the patient 14 was escalated to the dose of level 4. And then dose level, patient 14 to patients 16 were assigned to dose level 4 as the 4th cohort, while the 15 and 16 were under DLT assessment. Patients 17 and 18 were backfilled to dose level 3. And by the arrival of the patients 19, the cohort 4 on those level by the arrival of the patients 19, cohort 4 completed assessment with 0.0 out of 3 DLT. So therefore, the dose was escalated to dose level 5, and dose level 4 was open for backfilling. And patients 19 had similar conflict situation to patients 14. So accordingly, the patients 19 to 21 were assigned to dose level 5. And when the patients 20 and 21 were under DLT assessment, the patient 22 and 23 were backfilled to dose 4. And by the arrival of the patients 24, the patients 19, 20, 22, and 23 completed DLT assessment, but not patients 21 yet. And the dose level 4 for the cohort 4 had 2 out of 5 DLT, and dose level 5 had 2 out of 3 DLT. And we pull the DLT rate for the cohort 4 and 5, that will be 4 out of 8. So we de-escalate the dose to dose level 3, because also for the cohort 4, the DLT is 2 out of 5, which is also higher than the de-escalation level. And subsequently, the patients 25 and 26 were assigned to the dose level 3. And when the patients 27 was under DLT assessment, the patient 28 and 29 was backfilled to dose level 2. And when the 29 on the dose level 2 completed the DLT assessment, the state decision stopped the study. Okay, let's discuss the performance comparison between backfilling BOWIN and BOWIN, as summarized in Table 4. First, backfilling BOWIN generally outperforms BOWIN in Table 4, with a slightly higher percentage of corrected selection of the maximum tolerated dose in most scenarios, except for the scenarios where the true MTD is located at the highest dose level. And on the percentage of overdosing as a selection, the backfilling Boeing is about 2.6 to 4.0 percentage points lower than Boeing. And this is a continue of table 4. And compare with Boeing, the backfilling Boeing treats 3 to 12 more patients at dose lower than the DMT and allocates about 2 more patients at the true DMT and overdose slightly fewer patients. And more patients improve the reliability of toxicity estimation and enhance dose selection accuracy and provide better data for the phase 2 trials. And the average trial duration of backfilling Boeing is similar to Boeing design, and under typical patient recruitment scenarios, the backfilling does not prolong the trial duration. And this expansion of the active lower dose can be used to generate tolerability and activity data for dose optimization without compromising patient safety. And for the patients, the continuous availability of individual backfill slots allow efficient enrollment of patients who would otherwise be turned away under a standard cohort-based dose findings design. And that will be all my presentation. Thank you.
Thank you, Jolene. Are there any comments or any ideas about this trial design in this room?
Hal and Zolan, thank you for that. And that was a good explanation of a pretty complicated design. I was just wondering, could you tell if they do those simulations, are they always simulating a success? You know, did they do any simulations to say, oh, if this is a toxic drug, you'll find out, you know, what's the efficiency for that? Or it's sort of really based on the maximum tolerated dose? Well, and that could be a very low dose.
Yeah, and I think the first, so we have the probability, targeted probability, it was set by the clinician, and they may have some preclinical data before the drug was used on human. So the clinician or the pharmacist, they may use this data to choose the targeted probability, and that can make sure that the first dose or the lower dose is effective for the tumor. And I think that should be a explanation for your question. Is that your question?
I think so.
Yeah, and I think the all the simulations study they made was generating by the Monte Carlo simulation process. I think they have mentioned it in the supplementary materials. Yeah.
Are there any clinical areas that you think might be most benefited by this type of methodology?
And then also.
I think since this trial design most used for oncology, oncology trials, and I think maybe that's also like it still need time to check whether it is feasible or good to use since it haven't been widely used. But I think it will be most used in oncology trial, phase 1 trial to find the dose recommend for phase 2. Yeah.
Absolutely. Any other thoughts or so? I do have one question, and maybe those of you who are hoping to go into industry can weigh in, because this is something that I'm always curious about, how we decide which drugs move forward to phase 2. So let's say you were the one designing this trial. How would you package the results in a way that was interpretable? How would you explain that to the higher ups who actually have to make these decisions? Any thoughts or so?
I think so first we need to improve that it should be like effective for our disease or our target disease.
Yeah.
Yeah. And for the phase 1, we also considered about the safety data. Yeah.
Precisely. I think you hit the nail on the head. So we want to focus primarily, because it's phase 1, on safety, right? And trying to translate the results of this might be a little tricky, but I think hitting home the fact that it's either a safe drug and we should continue or not would be critical.
Yes.
Any other
Yeah.
thoughts? Yeah. Has anyone had experience in this process? We have a few more minutes if there are any other questions or so, or comments, thoughts on this methodology.
Well, one thing I quizzed both or pressed both of them on is, why is having more patients good? We always want to have fewer patients. Is there are there any scenarios where this would actually maybe identify a toxic drug earlier and have fewer patients exposed to it? Or too toxic drug, I should say.
Yeah, that's a good question. And I think the author they mainly talks about that the more patients will give us more information, and they are exposed to like the safety dose, and so we may not worry about like they exposed to the toxic dose. Yeah.
Okay.
But it did like increasing the funds that we need for the drugs. Yeah.
Any last thoughts? Okay. Well, it's 1 o'clock, so thank you all for joining.
Yeah.
Wonderful.
Thank you.
Great job. We'll see you
Thank
guys.
you.